So I'll start by introducing our speakers. Our first speaker is Siti Rahmayanti, MD, MMSc. She is a postdoctoral research fellow at program in Cellular and Molecular Medicine at Boston Children's Hospital. She earned her medical degree from Universitas Gadjah Mada, Yogyakarta, Indonesia, before continuing her master's in immunology at Harvard Medical School. Her current research interest is understanding the primary B cells response in both intrafollicular versus extrafollicular autoimmune environments. So welcome, Mira.
Hi, everyone. Thank you for having me. So happy to be here, and thank you for sparing your weekend with us.
Okay, so our next speaker is Marsha Wibowo. Marsha is from Jakarta, Indonesia, and is a fifth-year PhD candidate at Harvard Medical School in the Biological and Biomedical Science program. She earned her bachelor's degree in biology at MIT in 2016. She's passionate about utilizing computational tools to gain meaningful understanding of disease from big biological data. Her current work focuses on the gut microbiome and its relationship with metabolism and diabetes. Outside work, Marsha likes spending time with family and friends, playing board games, eating Indonesian food, and giving back to the community. Her dedication to Southeast Asia led her to serve in the Southeast Asian Service Leadership Network, or SILNET, for 8 years, where she organized service and leadership projects in different Southeast Asian countries and held various leadership positions as co-president, professional mentor, and board of directors. So welcome, Marsha.
Hi, everyone. Yeah, thanks for having me here and excited for our discussion tonight.
I'm also excited to hear from you. And our last speaker is Professor Arif Satria Wira Kusuma. He is an assistant professor in the Department of Biological Pharmacy, Faculty of Pharmacy at Universitas Pajajaran, Unpad. He received his Bachelor of Pharmacy degree at Universitas of Pajajaran in 2012 and Master's degree in Biotechnological Pharmacy at ITB in 2014. While taking a faculty position in UNPAD, Professor Arif worked with USAID to facilitate student exchange between UNPAD and Rutgers University of New Jersey and acted as visiting scholar in the Department of Pharmacology of RWJMS, Rutgers University. Professor Arif is currently pursuing a PhD program as a Fulbright Fellow in Dr. Huicho Fan Lab of Rutgers University, studying GRGA, a novel transcription factor from pathogenic Chlamydia trachomatis. He is looking forward to being the first US graduate in the Faculty of Pharmacy at UNPAD. And during his time in the US, he's hoping to promote Indonesian cultures and build strong collaborations between US and Indonesia institutions. So welcome, Professor Arif.
Hello, everyone. Thank you for joining this event. Looking forward to our discussion.
So again, thank you so much, everyone, for being here. Yeah, so with that, I guess we can start by sharing like the speaker stories. So from Mira, from Mira, can you briefly elaborate how you got here, how you got to where you are now as like a postdoc?
All right. So basically, my starting point from where I was before, which is a full-time practitioner. So I was practically working as a doctor before I came here. So my work was way far from what it's like from asking biological questions. And I assure you guys, not as cool as my two friends here, which is Marsha and also Prof. Arif. I think they've been consistently in this past before, but I think it's been like a very fulfilling experience for me, starting off by attending a master program here at Harvard Medical School. And then luckily during that program, I think the benefits that students have is actually the hands-on experience in the lab. So we are directly connected with a lot of faculties, and we're even getting the experience to actually make our thesis, build it from the scratch, and actually performing what it's like to be a real scientist. So that was my starting point, actually my master's program, which I finished last May. And from that, I was offered a position with one of my professors in the program, which is one of the boards in the immunology program. And he thought that I might be interested in the position because it's related to the realm that which I was interested in, which is the autoimmunity. And yeah, we had a discussion, and I have worked there from last August, actually. So I'm pretty thankful that within this chaotic pandemic, I still can find, I can still secure a position where I enjoy myself to work within. So yeah, that's how I got here, basically.
Wow, that's interesting. So you started as a doctor and then you got into research up there.
Yes.
How about Marsha? Would you share how your journey until here?
Yeah, I guess I started becoming interested in biology back in high school. So I started joining biology competitions and the OSN and IBO, International Biology Olympiad, and because of that, I think I got exposed to a lot of just trainings and biology, and that made me even more interested in doing biology. Initially, I wanted to be a doctor, but that path was kind of burned, and I guess I didn't. I wasn't able to I didn't get admissions into schools in Indonesia because I wanted to be a doctor back home, and so I had to take a gap year, so 1 year before I applied again to other schools, and that's when I decided to apply abroad, and I tried applying to schools in the US, including MIT, and luckily I got accepted into MIT, and that, I guess, yeah, I decided to go there, and I majored in biology, so I came to the US back in 2012, and I think at MIT, my eyes were really opened to the different possibilities of doing research, and I was able to be involved in research projects with the professors there. There were just a lot of opportunities, and because of that, yeah, I decided that that's what I wanted to do. I wanted to do research, and I decided to apply for PhD programs, and yeah, that brought me here, I guess.
Oh, wow, that was amazing. So Marsha couldn't be a doctor back home, so she went
and then
to
go
US.
to MIT. Such a nice comeback.
Right. Yeah, how about Professor Arif? You want to share your story?
Sure. Okay, so after graduated from ITB in 2014, I applied as a faculty member in my alma mater as an undergraduate student, which is the Faculty of Pharmacy, Universitas Pajajaran. Then I got accepted. And then starting from 2016, I start to work with Rutgers, in which in 2016, we got a 3-year grant program sponsored by USAID. And the program is called Research Internship Fellowship, in which every summer for 3 years, we accept master students from Rutgers to do research in Indonesia, especially in Universitas Pajajaran. We did a lot of different research back in 2016 up till 2018, including testing anti-cancer effects from indigenous Indonesian plants. And then we also did research about tuberculosis in Jambi area because it was it has pretty high case rate there in Jambi. So we work with our partners in Universitas Jambi and Rumah Sakit Raden Matahir in Jambi. And then by the end of 2018, Rutgers seems to like me, so they did offered me 1 year visiting scholar programs to come to New Jersey, and then I took it. I worked with Professor Huizu Fan, which works on Chlamydia trachomatis, and he did study about the novel transcription factor. And then during my visiting scholar programs, I also tried to apply for Fulbright fellowship, in which I got accepted, luckily, because it was a pretty long and complicated process. And then I started my PhD on molecular bioscience in fall 2020. So I'm a first year student right now, and then I'm on my second semester. I haven't started my lab rotation yet. I will start it next month. Yeah, that was my story. Thank you.
That's quite a story. So you are both a professor and a PhD student.
Yeah, that's right. That's right.
So I guess moving on, can you guys tell us a little bit more about the kind of topics or things that you are researching currently? I guess like we already touched a little bit on this, but maybe yeah, you want to know a little bit more. Maybe starting with Mira.
So yeah, so my lab in particular is interested in the autoimmune B cell reaction. So basically, when we're talking about autoimmunity, or maybe I should start. I'm not really sure how exposed you guys are with immunology, but basically, within our body, the immune system is supposed to be able to tell the difference between our self and non-self, which could be something they're detrimental, such as viruses, bacteria, or the stuff they're basically our common enemies. And within that process, sometimes there's kind of like an error where somehow our body is turned their backs against us and they kind of like attack ourselves. And that's just the basis of autoimmunity. And in this orchestrated situation, actually there are a lot of players in there. There are T cells, there are B cells, and a lot of different kind of immune cells that you might not be interested in. But anyway, my lab focuses on the cells that called B cells that is important in producing antibodies, and also they are also important in producing some sort of memory cells, which in secondary kind of attack, they will act very rigorously, faster, and more effective. Basically, when we have this B cells, they are autoreactive, means they are actually attacking ourselves. We would like to know what drives them to act this way. We want to know what caused the breaking of the tolerance, because we're supposed to be able to knock these cells down before they get uncontrolled, right? So basically, when the B cells is being developed within our body, they will have these checkpoints where they are being killed if they are those people or like those cells, they are actually attacking our own cells. But somehow they escape this tolerance mechanism. So we want to know what causes this to happen, what caused this phenomenon to happen. And not only they attack certain address, what I mean as address here is that there's a term called epitope. Epitope is like something that you recognize on like foreigner. For example, like Susan. Oh, Susan has a nice earring in her ear. So our cells know that Susan is dangerous and Susan has that earring. So whenever the cell sees that earring, he immediately recognizes that it's Susan, and Susan is going to be like destroyed. Sorry, Susan, but practically something like that. But during this autoreactivity mechanism, this autoimmunity, not only the cells is attacking the earring, they also start to attacking that flowers on the sweater, or even attacking other people. Like, for example, like we have here Marsha. Oh, Marsha has a long hair. Why don't we just attack her as well? So, and this mechanism causes a lot of problems. because within our system, there are like different organs, which is also harbor these cells. They're unfortunately being autoreactive. So these organs and tissues are being attacked everywhere, and that causes the people with lupus become very sick. They start to having problems with their heart, with their kidneys. So yeah, basically, when we talk about this medically, we only like seeing it as, oh, there's a problem with the kidney, so we have to treat the kidney. And then if you see a problem with the heart, then we have to treat the heart with the ampoules of drugs that we have. But as a scientist, now I see it differently, because then I got to ask a different question. I have to ask deeper questions like, oh, why the cells are behaving like that? What drives them? What kind of like nutrition they get? What kind of metabolic changes that happens in there? So yeah, so basically that's that's sort of questions that my lab is trying to seek. So I hope that's not too confusing for you to hear, but yeah, basically that's what I study in everyday situation right now.
Okay, so that sounds a little bit, that sounds very complicated, but that's definitely a layman, like a good way to explain it. I mean, full disclaimer, I guess I am by no means know anything. I mean, know a lot of things about life science. My background is math and computer science. So yeah, but I understand a little bit of it. So that's good.
Okay. Thank you.
So I guess moving on, how about Marsha? What research or what topics are you working on?
Do you want me to show the slides now, or is this just a bigger picture?
Yeah, you can show your slides.
Okay. Sure.
I think you can share your screen. It's enabled. Yeah.
Okay. Can you see my screen?
Yep.
Yeah. So my lab, I'm co-advised by Alexander Kostic and Yuhua Tang, and I'm based in the Joslin Diabetes Center. So it's a diabetes clinic, but we also have a research part of the building. And so coming into grad school, I was really interested in studying diabetes, but I learned more about the gut microbiome. Some of you might know about it, some of you might not, but for me, it was something new that I learned a few years ago when I started grad school. And basically, those are the bacteria that live in our gut, in our digestive tract, the good bacteria that could help us digest things and all kinds of stuff. And there's been a lot of studies that show correlation between specific gut bacterial species and our metabolism, including diabetes. And so that's kind of the big picture of my lab, what my lab does, gut microbiome and diabetes and metabolism. And so for me specifically, this is the main project that I've been working on for my thesis. The title is Reconstruction of Ancient Microbial Genomes from the Human Gut. And again, just a little bit of background. So diabetes and obesity and other chronic diseases have been increasing dramatically in the past 60, 70 years. You can see here the number of people with diabetes, this is in the US, has been increasing dramatically. And one factor that might play a role is actually the gut microbiome. So the gut microbiome are the trillions of bacteria that live in our digestive tract, and they're important for various things, including metabolizing nutrients, maintaining our intestinal cells, and protecting us against pathogens, and modulating our immune system. But when the composition of this gut microbiome is not normal, then a lot of like bad things can happen, basically. So here, for example, it's been shown that industrial populations, so these are populations like people who eat a lot of junk food, like who use a lot of antibiotics and who live a sedentary lifestyle. So industrial populations have a higher incidence of chronic diseases, so more obesity, more diabetes. And at the same time, research has shown that these populations also have a lower diversity in the gut microbiome, so fewer species in their gut microbiome. Whereas in non-urban populations or traditional populations, these people are usually more protected from chronic diseases, including obesity, and they actually have a lot more bacterial species in their gut. And so one possible treatment for obesity or diabetes is to reintroduce some of these bacteria that are missing in industrial populations, and that could help with this increasing trend of obesity and diabetes. But another source of good bacteria could be from the past, or what we call paleophysis. So to study these gut bacteria, you can take, this is basically fecal samples, poop samples from the past, like 1,000, 2,000 years ago, that have been dried out and like preserved. So the bacteria are preserved in these feces, and the bacteria are not alive anymore, but you can take their DNA, you can sequence the DNA and figure out what bacteria are there. And maybe there's a lot of other bacteria that have been lost in the modern day, in the present day. So that's basically what I've been doing. So we worked with some archaeologists who collected these paleophyses, ancient fecal samples from the desert in the southwestern US, and we sequenced, we took the DNA from these samples and sequenced them. This is a highly specialized method, like you have to do it in an ancient DNA special room, because everything has to be super clean. Like you can't have any contamination, because we don't want to contaminate with modern DNA. And from this, you can figure out the bacteria that are there, and also you can assemble like these, I hope this makes sense, but I don't know if this makes sense, but these raw, like these short sequencing reads, you can assemble them computationally into longer stretches of DNA, or what we call contigs, and then you can figure out what genes are in these contigs, and you can, what we call, bin them. Basically, you compile the contigs that belong to the same genomes, and you can kind of make these like draft genomes. So you can, from these short reads, you can make draft genomes, even like almost complete genomes, bacterial genomes. And this has been like really challenging, especially with ancient DNA, with ancient feces, because a lot of the DNA is like highly degraded over like 1,000 to 2,000 years old. There's a lot of degradation and DNA damage. So it's been really hard to do this. And this is something that we've been able to do in our lab recently. And so from these samples, we've been able to identify microbial species that are lost in industrial populations, but present in these paleophyses. And we were able to reconstruct hundreds of ancient bacterial genomes and find novel species. Also, we've discovered genes and pathways that are enriched in the paleophyses, and really just better understand the evolutionary history of our gut bacteria. And like, hopefully that will really help us restore our health in the future. But yeah, so that's kind of what I've been working on. This is a paper in progress, but it's almost near completion. So yeah, I just wanted to share that with you all.
That was really interesting. Never would have thought like the bacteria in your gut affect that much of your health. Yeah. Um, so I guess moving on to Professor Arif, do you want to share more about the topics that you are researching currently?
Sure,
Or
sure. I will
even
try
your
to
past
share.
projects? Yeah.
Okay, I'll share my screen. Okay, can you see it? All right, great. Okay, so as I told you earlier, that I work as an assistant professor in Universitas Gadjah Mada. I took my undergraduate and pharmacist professional academic school in Universitas Gadjah Mada, and then I took my master degree in the Department of Pharmaceutical Biotechnology in Institut Teknologi Bandung, and then right now I'm at Rutgers as a first year PhD student. And this is my stats in Scopus, which I currently have 12 papers published. And then, as I told you also earlier, that the collaboration of UNPAD and Rutgers started on 2016. For 3 years, we accept master students from Rutgers to do research. This picture was taken in Jambi, in which we were visiting one of the patients for multi-drug resistant tuberculosis. We tried to do interview on how they took their medicines, and then we did also analysis in different genes related with the multi-drug resistant tuberculosis. And then this is my past research. Maybe I can correlate with Marsha a little bit for my first topics also about gut microbiota. In this study, I used animal model, which is the adult male New Zealand rabbit. And then it is a collaboration project in which I work with Professor Ingrid Surono from Department of Food Technology in BINUS University, Jakarta. By doing this, we wanted to see the effect of giving probiotic to animal model, whether this probiotic will enhance the absorptions of the drugs that we give to the animals, and then also other side effects that we kind of see, we wanted to see is whether the probiotics also been able to reduce the pathogenic population bacteria within the gut. And this is the papers that we managed to publish from this study. At least 4 papers from 2 labs in Universitas Gadjah Mada, which is the microbiology and biotechnological laboratory, and also medicinal chemistry laboratory. And this is the result example of the study that I do, that the supplementation of probiotic Enterococcus is able to decrease the population of non-beneficial Enterobacteriaceae. This was published in 2019. We did several different tests to get to these results. We combine the conventional and also the nucleic acid, which is we know right now probably everyone is pretty heard this PCR thing a lot. We also did PCR on bacteria to determine the 16 sRNA as the genomic identifier for Enterobacteriaceae member. And so mostly my research topics is related with infectious disease, which caused by bacteria, and also biotechnology. This is my second project, which I did back in Indonesia. We work with pneumococcal vaccines, in which in our country, the pneumococcal vaccines up till now is not yet included into the Program Imunisasi Nasional. So by doing these studies in collaboration with a lot of partners, such as Clinton Health Access Initiatives and also Sabin Vaccine Institute, in the Washington DC. We wanted to make our result be considered by the central government, in this case by the Ministry of Health, that the pneumococcal vaccine is necessary and have to be included into the national vaccination program. And we did this back in Lombok, in which why we choose Lombok is because they have established a cold chain system as a pilot model for different area in Indonesia. Lombok is a small island, so making Lombok as a pilot study for our study is one of the reasons that why we choose Lombok as a location to do our pilot study. And then based on these studies, right now we haven't yet published our result, but we do already have 2 review paper published in Scopus. And then by publishing this, I mean, if you are interested in learning more about this, you can see that the mathematical model is also relevant to this field of studies, in which we suggest that by predicting the vaccine distribution using the mathematical model, we can give a good result about the suggestion. Oh, sorry. And then also this one was published between Unpad and Rutgers. Yeah, this one. The first name, Fonet Fonjungo, is a student, master student back in 2018, visiting Indonesia. Now she's in a medical school in Mehari Med School. And then during her visit in Indonesia, we wanted Fonet to do research about sustainable financing for new vaccines in Indonesia. What are the challenges and the strategies? And then the last, the topic that I did study when I'm at Rutgers from December 2018 until 2019, I did about sexually transmitted disease related studies, in which I study about the novel transcription factor from intracellular pathogenic bacteria, the Chlamydia trachomatis, which has gene regulator A, or abbreviated as GREG A. So this is, as we know that in the United States, the CDC have published statistical data that in 2018, more than 1.7 million cases of chlamydia were reported to CDC from 50 states and the District of Columbia. But it is estimated to have escalated like annually until it reached the point of 2.86 million infections. So the use, currently the treatment of the chlamydia infection is using broad spectrum antibiotics, in which it is not a pretty good choice because it can leave non-beneficial side effects, such as killing other good bacteria within our body when we use the broad spectrum. So by identifying this specific transcription factor, we wanted to make specific drugs that target this transcription factor. So then in the future, maybe we can replace the use of broad spectrum antibiotic as a treatment for chlamydia infections. Yeah, and why I chose to join this lab during my visiting scholar period is because it is an intracellular pathogen, despite we in Indonesia does not have high case rate of Chlamydia trachomatis infections. But by studying the Chlamydia trachomatis, I was hoping to be able to learn the technique that in the future I can apply towards other intracellular pathogen, which is one of them that I targeted is the Mycobacterium tuberculosis, which is still the rate of infection caused by Mycobacterium tuberculosis is still very high in our country. So that yeah, that's why I am interested in studying Chlamydia trachomatis. But in the future, I may or may not continue this study because in the second semester here in Rutgers molecular bioscience program, we first year students are required to take lab rotations in 3 different labs. And then for each labs, we have to spend around 6 to 7 weeks. And those 3 labs in the end, maybe will be my choice of lab to spend the rest of my PhD study time. So we'll see what other chances that I have here while I'm in Rutgers. I think that's all that I can see. We can go back to you, Susan. Thank you.
Thanks, Prof. Arif, for sharing. Those are some great work. Some of them are very interdisciplinary too. So, wow. Yeah, so I guess we can now move on to the Q&A session. So I would like to remind all participants that if you have any questions, you can type in your question on the link. I think it's in the chat box. You can also upvote, or yeah, you can also upvote other participants' questions if you have like similar questions. So we already have some questions here. First, to Professor Arif, what do you think is the biggest challenge in doing research in Indonesia? Is funding a big issue?
Okay, so I have to answer this in different perspectives, at least 2. First, from a professor perspective, for professors, the funding to do research can come from different sources, such as the ministry, and then the university, or also other third parties. Like what I explained before, during our pneumococcal vaccine studies, we got sponsored by the Clinton Health Access Initiatives, which is based on the United States. And why they wanted to do this is that they want to give back to the community. A lot of big companies here, I know that they do this kind of actions, which is giving back to the community. So the second perspective is from the student's perspectives. As a student here, as a PhD student, I don't think that we worried about the funding to support our studies, because most of the professors have already had their fundings either from NIH or NSF or from the university itself. So what do we really need as a student to come to the United States? Plus, the other factor is that PhD position here in the United States is a paid position. So by applying to Fulbright, I managed to secure that part, that being responsible as my part. And then yeah, either way, if you don't have any scholarship or fellowship at all, it doesn't matter, because you have the chance to also be offered a position as a research assistant or a teaching assistant, which is also support your payment during your PhD studies here in the United States.
I see. So in terms of companies giving back, I guess as a follow-up questions, do they usually give back in like what kind of fields? Do they usually choose like the fields that are similar with where they are operating, or this can be like totally like unrelated? I'm just curious.
So I didn't have the experience to receive sponsorship to do research from other than Clinton Health Access Initiatives. But as we know from the name, that they focus on the health-related field, so that when we apply to Clinton Health Access Initiatives, we explained that in Indonesia we have a major problem, which is the pneumococcal vaccine is not yet implemented nationally, and it is not yet included into the national immunization program. And we need to do studies to urge the government to put this vaccine into the program, because right now we face another effects of not implementing the PCV into the national program is that we have high rate of stunting in our children in Indonesia, which is in the next 10, 15, 20 years will also contribute to the GDP of our country, because children with stunting are predicted to not have proportionally good growth rate in terms of height, in terms of brain capacity. A lot of studies have revealed this, and we need to address this issue from the top. That is how we try to explain to the Clinton Health Access Initiatives.
Gotcha. Yeah, thank you for the answer. Yes, next question. To Mira, what is the most challenging aspect in immunology research in Indonesia compared to in the US?
I think I would echo what Prof. Arif had explained, that of course the main difference will be the funding. I think as what he mentioned, that here as a budding scientist, I would say that we're still trying to expand our technique in research, as well as trying to find mentorship during our training here, to hopefully will help our career in research fields. And in my case, probably it's also my goal to hybrid both part in the clinical side as well as the full-fledged research field. But yeah, so I would say I think here the system in which how funding is distributed is totally different. So it's, for example, here in Boston, there are more than 500 PIs securing probably 50% of the whole NIH budget. So you can imagine how limitless is the potential that you can experience if you manage to have the opportunity here. And as also what he already said, that there's so many different kind of positions that you can apply for here. Not only in terms of fellowships, you can also get a degree in at the same time. And so I think this is something that is very attractive, not only for the Americans, but also from everywhere, everyone around the world. And what I want to emphasize here is that to Indonesian students who are maybe interested or even remotely interested with life sciences, I think this is one of the destinations that you would want to consider. Why? Because of that even only for that reason, because I think we still need to increase the number given our great number of population. And I truly believe that the Indonesians have the ability to also compete and expand. And hopefully, I'm not pessimistic that in 10 or 20 years, I mean, Indonesia has been evolving very fastly. So I think in the 10 years, it's not impossible that we can replicate what is happening here. And if more people are exposed and more people are being given the opportunity, I think we can have a better dynamic back home in Indonesia, where we can have more third parties actually supporting science, and more companies are contributing. The government are more aware of the importance of the chain, because not only you need the research core in the university, you need the industry to back up, so then everything can comes in a first circle. So to sustain that research practice, you need that chain to fulfill. And unfortunately, it's not available and it's not thriving yet in our country. So I think the biggest obstacle, I would say, is still the funding, and also the core and the facilities that we have.
I totally agree. It's definitely a chicken and egg problem. Like, you need more scientists to create a good ecosystem, but you need a good ecosystem so that there are more scientists. Yeah. So next question is for Marsha. So the question is, why are you even remotely interested in studying poop? I guess.
I was looking at the questions, and I just have to say I love all the questions. Like, these are very, very good questions. Yeah, I saw that one, and I think it's actually very cool. Like there's actually like a lot of things you can learn from your poop, and as I was saying, like there's, I don't know, like trillions of bacteria in our digestive tract, and they come out in our poop, and there's like more bacterial cells in our body actually than human cells, and there's more bacterial genes in our body than human genes. So they play a big role in our health, and there's a lot you can learn from them. And I think even they can weigh, like the gut microbiome can weigh up to like 5 pounds, which is like 2.3 kilograms, which is, I think, heavier than your brain or your liver. So people have really started to think of the gut microbiome as another organ, and really like a big part of yourself. And yeah, just other studies that have shown how much they can play a role in diabetes and other diseases. For example, when you basically transfer poop from like a healthier human to like, or even like mice or something, for if you transfer poop from like a lean individual into a more obese individual, like you can make that individual lose more weight and things like that. That I think is just fascinating, and just being able to understand better what some of the key players in this, I think it would be very, very, very cool.
Yeah, that sounds cool. I guess next question also for Marsha, still about the research. So if you're trying to introduce past microbiomes to the current human stomach, given that people have shorter lifespans in the past, how do you know they don't have any bad effects?
Yeah, I think that's so what my study has shown is that we can learn about the genetics of these microbes and how much they've changed in the past compared to now. But to get to the point where we can introduce these ancient bacteria into humans, that requires a lot more research. Like, we'll have to test these in mice. We'll have to see the effects on the host physiology and all kinds of stuff. And so that's going to take time for sure. And the other thing is, those bacteria are in the samples in the paleofeces, like those bacteria are not alive, so they're dead. So we can only know like what the DNA sequences are, but we can't like culture them in the lab to like bring them back to life. So a more possible approach, I think, would be to figure out like what genes are important in these bacteria and like put those genes back into modern bacteria with like CRISPR or something like that. Yeah, that makes sense.
Can I ask follow-up question? Because I'm very interested, Susan.
Yes, definitely. Yes.
Okay, Marsha, with your study, can you spill a bit of tea about how similar or how different it is from our current species with the fossil species. And the second one is fecal therapy has been used in medical world for, I think, for a decade's time. But how do you think it correlates with the gene itself? Because would you think it's like something somatic, or it's more like the environment that actually make these lean people lean, and hence also causing drift in their microbiome population?
I see. So your first question is how different the
and similar, or maybe how similar or how different.
So I compared like these ancient feces with hundreds of, like about 800 modern feces from like all over the world, and basically the pattern that came up is like the people from traditional populations. So I analyzed some data from, I think it's like Tanzania, and then there's some traditional populations from Mexico. So those populations look a lot more similar to these paleofeces compared to, let's say, the gut microbiome from like the US or even like Europe and things like that. And there are a few species that are completely missing in the industrialized population. Like we don't see it at all in the US, in China, in Europe, in like industrialized populations, and but they are present in all of the paleofeces and a lot of the traditional populations. I think I don't know if I should go more specific than that. Maybe like wait until the paper is out, but yeah.
So they understand again. And for the second one, for what makes these lean people has that kind of fecal population or microbiome population?
I think I personally think a lot of it is diet, because like the gut microbiome changes a lot even throughout the day, like depending on what you eat, like it changes a lot. And diet, like fiber, like low fiber is really bad for your bacteria, because they basically don't have anything to eat, because they eat whatever we don't break down. So like fiber our bodies can't break down fiber, so those become nutrients for the bacteria. But if they don't have those fiber, then they start eating our mucus, like the mucus that lines our intestinal cells, and like those have like various bad effects. So I think like diet plays a big role, honestly. There are some studies that show that genetics also can affect the gut microbiome, but yeah, I think especially for obesity, diet definitely plays a big role. Yeah. Thanks for the questions. Yeah.
So the lesson is you should eat veggies for our bacteria. Okay, so next question is for Professor Arif. So how do you create an elaborate team that consists of health economists, doctors, researchers? Do you collaborate with Dinas Kesehatan to help secure the vaccine to undang-undang?
Okay, so yeah, back in Indonesia, I think it's pretty doable to do this kind of multidisciplinary research, in which I came from a university background. I work as a faculty at Universitas Gadjah, which we have at least 5 schools related to health. We have the School of Medicine, the School of Dentistry, and then the School of Pharmacy, and then the School of Nursing, and the last is the School of Psychology. And this gives us the chance to use the resource within UNPAD community. We try to communicate with faculties from different schools that we have this undergoing this project currently done, and then to be able to advocate this kind of results to the health ministry. my PI have been selected as one of the member in ITAGI, which is the Indonesian Technical Group Advisory for Immunization, so that he has the power kind of to sound about what we found actually in the field during the pilot studies in Lombok, that it is possible for us to put the PCV or pneumococcal conjugate vaccines into the national vaccination program.
Right. So, would you say, I guess you wouldn't know how it is here, but would you say it is harder here than it is than it is in Indonesia, or like, are they very different?
I cannot say like it is harder here because I don't have the experience to work with other field. During my visiting scholars program, I only work in kind of 2 or 3 labs, but these 3 labs are in the same departments. So that is also one of the strong point that we have here in the United States, that we can use different equipments from different labs as long as we communicate good enough, and then the professors who own the lab allowed us to do so. Sometimes I found this to be hard to do in Indonesia, in which labs are closed only for the labs members. So yeah, this kind of thing kind of make me learn more about what is the big difference of doing research here in the United States and in Indonesia, and each has its own advantages and disadvantages.
Gotcha. Yep. Thank you, Professor Arif, for the answer. Next question for Mira. So after this, I guess for the future plan, are you planning to be a doctor back in Indonesia?
Yeah, for sure. I think once my training is done, I hopefully can somehow implement what I have learned here. And I hope I can still practice some of the basic science that I have, because I think now some centers in Indonesia has started to actually build their own research core. So both in, like the national state university, as well as some of private universities. So I'm hopeful that I can actually practice and implement what I have learned back in Indonesia.
Yeah. Maybe you'll also have your own lab later back home.
We'll see.
Okay. Another question for Kak Mira. Hi, Kak. So this is from Jessica. She said, I'm really interested in your study. How will immunology studies be applied in Indonesia from your point of view?
Okay. So in Indonesia itself, I think now we are going through a double whammy situation where, like what Professor Arif now studying, that a lot of infectious disease like tuberculosis and then different kind of infection rate is also very, very high, which practically already very most likely diminish here in the United States or other developed countries. Now Indonesia, as with the change of diet, with the change of lifestyle, we also facing a lot of non-communicable diseases problems such as diabetes. We have a lot of increase of autoimmune cases. And this problem is a very problematic because now we don't really have the definite cure for a lot of these diseases. They are chronic diseases, mostly in productive people. And of course, it will impact a lot for those who are still in their active years or who needs to be working, also giving a lot of impact to the economy with high expenses. For example, here in the United States, for each year, each patient needs to spend around $50,000 for SLE therapy. And a lot of this therapy is not accessible yet in Indonesia. And in the future, we're not only dreaming of like a generic therapy where we're just try to suppress the immune system. For example, the use of a steroid. It's how steroid works. It's basically suppressing your immune system. In the future, we hope to actually target certain receptors or even certain protein that might be able to improve the disease activity. So that's why I think this kind of studies will come in place, and how important it is to actually be in the front runners, even though maybe now we're a little bit behind. But I think once we have all the weaponaries, we have all the technologies, then we should be good to go. We hope to be able to contribute. As far as I know, today, from data from last year, I think each year we have around 5,000 to 10,000 cases. But again, this is mostly concentrated in bigger cities, and we have so many populations that are still unreachable. They don't have access to healthcare. So this number is very much, I think, understated. So I think we still need to focus more on making better diagnosis for the patient. Yeah.
Gotcha. Thank you, Mira, for the answer. So next question is for everyone. Does the research idea come from like yourself, or is it from another ongoing project? How do you address your interest to be in line with the funded research in the lab? I guess maybe Marsha can take this first, then you can go.
I mean, I guess it depends on the lab you're in and your PI, but for me personally, I was pretty clear in the beginning that I wanted to do something related to diabetes, but then I also wanted to learn computational biology. So doing the computational techniques, and I communicated that to my PI, and so he was just he was just approached by this archaeologist that they have these ancient fecal samples, and they were like, "What? We have these. Like, do you want to do anything with it?" And so I was there in the initial meetings, kind of talking to the archaeologists and like thinking about like what can we do with these samples. So the samples, for me, the samples kind of came first, and then we kind of thought about it together, like what would be the most interesting question to ask with these precious samples, and that's how the project kind of evolved, and throughout the way, I guess, I think it's been, yeah, like both of us developing the ideas together and like guiding the research together. Yeah, I think for me it was kind of very collaborative, like yeah, that's my experience.
How about Mira and Arif, Professor Arif? Do you guys have anything to add? A bit similar or not?
Yeah, I think I would agree with Marsha, because I think it really depends on the lab. I have experience in 2 different labs, and trust me, sometimes you have something that you're really interested in, and sometimes it's not aligned with your PI, and that's okay. You have to just keep it in the back and do what is, you have to compromise, and somehow you will align your interest with what is available in the lab, because different labs also have different, I would say, different technique that they use. So not always implement, you can not always implement certain technique that you have in certain lab, and that's how you grow, I think, as a scientist, that you learn from different people and then you adopt their technique, because different people sometimes do things differently as well. And there's a lot of variables that relate to it. In my experience, we started off with the big team that we both are agreed upon, which was the autoimmune part. And then from that, we kind of like talk about the study that has been going on in the lab. And some of these studies are unfinished. So I was kind of like a fill-in in that in the beginning, but then as the research progressed, I also find things that I'm personally interested in, and that's how I built upon my next project. So I think there are different ways of how a research project started, and it could be very different with different people and how the circumstances surrounds it. At least that's based on my own experience.
Professor Arif, anything to add?
Yeah, sure. I do agree with both Mira and Marsha, in which at the beginning I was pretty clear that I set my umbrella. I want to do research in microbiology and biotechnology. And then during the PhD application process, I try to see which faculty have worked on this kind of projects. And then even though in the past I never worked with chlamydia, but then I see what is the professor is trying to do with the studies in chlamydia. And then from there, I decided that this is going to be my taking home point, that even though I will not continue, for example, to do research based on chlamydia, but I know that intracellular pathogen have similar features that I can apply towards more like hot issue in Indonesia, for example, in tuberculosis or other intracellular pathogens. That's how I approach on deciding which projects that I wanted to take on.
Okay, gotcha. I guess the next question is another one for Professor Arif. Is the if you're a professor currently, then is it common to do a PhD, or like why are you pursuing a PhD as a professor?
Okay, so I think I'm the last batch from the Ministry of Higher Education that is accepted as a faculty position with a minimum requirement of a master degree. So I was accepted in 2015, and then the next year the requirement is increased, in which to be able to apply as a faculty position in either state university or a private university in Indonesia, you have to at least have PhD degree. So during that point, 2016, Universitas Pajajaran made all their faculty to be either registered or currently trying to apply to different PhD programs. And that's how I ended up here.
Gotcha. So I guess just a reminder for all the participants that you can always you can ask the question on the link. You can also upvote other people's questions if you like them. So thank you, Professor Arif, for the answer. The next question is for all speakers. When applying for a PhD, how do you relate your background to fit your PI research topic? Do you choose your own topic, or do you need to follow everything that they ask? I guess this is pretty similar with the previous funding and lab questions, but if you guys have anything to add for that, please do so.
I think just the first part of the question, like when applying for a PhD, how do you relate your background to fit the PI research topic? I don't, at least from my experience, I don't think that's necessary, especially for PhD programs in the US. people are under the assumption that you're going to learn what you need to learn during your program. So for me in undergrad, I never worked with the gut microbiome. I didn't have experience with like computational biology. I just had research experiences for 4 years. Like I worked in like immunology lab and developmental lab. So I've done research and I know that I wanted to do it. But I don't think it's necessary to like fit, have those background knowledge before you start your PhD about the specific topic you want to research. Yeah.
Just a little bit addition to Marsha. Also, other important factor on deciding which labs are you joining in is, on my opinion, is also based on the PI, who you want to work with for 4 or more years to finish your PhD. Because if you are not comfortable in the beginning, I'm pretty sure that it will give you a lot of trouble in the future.
I agree. Yeah. Also, I think the system is very different in the US. I think what the question is referred to is maybe like more appropriate if I know that some of my friends who do their PhD in the UK, they actually apply to the lab and then they actually do the work and get a degree while working within the lab. But in the US, it's actually like what Prof. Arif said. I think in the first year, it's like a basic foundation year. So I went during my master, I was in the same class with the PhD students because they had to actually take those basic classes on immunology. So then I think they actually figure out what they want to do after that foundation year, and they start to do the rotation. So they get exposure from different type of labs, different PIs, and maybe the sort of project that inspire them. So I think it comes with the notions of those American dreams team. In America, I feel like it's just a very different nuance than what's in the Europe, I think, because here they really want to know you as in person. They want to know your personality, whether you can get along with your PI. They want you to be comfortable. Some students even drop the lab after 3 months, and that's normal because they don't think that's going to work and they want to switch lab. It's totally normal to do that here in the US.
Right. So to summarize, is it correct to say that in the US you apply to the program and don't need to apply to a specific PIs, but in other places you might need to align your background to fit a topic? Right. Okay. So I guess moving on, this is probably going to be the last question. It's for Mira. What is the most common autoimmune diseases and allergies in Indonesia, and how far? So yeah, how far is the government concerned about it? If yes, how far are they concerned about it?
I think for the actual epidemiology, it's very understudied, unfortunately. So I don't think we can come up with a real answer here. But as for my experience as a practitioner myself and a lot of discussion within the community, I think a lot of people are actually now diagnosed with SLE. Even my own aunt has SLE, and psoriasis is also a lot in the dermatology clinics. And as for allergy, I think the most common is dermatitis, and also some people relate rhinitis with asthma as well as bronchitis, especially in the younger population. So it's actually pretty common to find the cases in the clinics. And sorry, what's the follow-up question again? I think just the most common ones, right?
Yeah, and like what does the government do about it?
Yeah.
Yeah.
I think as for autoimmune disease, I think that more debilitating are those who are more systemic, like SLE and then MS, like sclerosis and psoriasis, because this really affects the livelihood of the patient. And I think people are still very lack in information in regard to this stuff, and we also have very few experts in the field. So, for example, to have an appointment with certain consultants in this field, it requires like a long period of time, and waiting time is also is very I think is very hard for the patient as of now. And also the option of therapy is not as easy, because the more targeted therapy is usually not available yet. So I think the patient is still usually being managed when they're having flare only. For example, if they have like a serious high increase of creatine, for example, because of the failure of kidney, and they will get the therapy for the kidney, but not the actual disease. So I think in the future, I hope we can address these problems more, and hope the government will also kind of highlight this issue more at the table for the general population.
People are more aware about this kind of diseases.
Yeah, I think the awareness is not there yet. Yeah.
So okay, I lied. This is the last question. That was not the last question. So I guess for everyone, for those of us who are interested in like doing graduate school in life science or related fields, do you have any tips or suggestions for that? Maybe Mira first.
Me again. Maybe Marsha answer for this one.
Professor Arif, maybe.
Okay, I mean, for those who are interested in doing PhD in life science in the United States, I mean, don't worry. I mean, a lot of people try to really sweat out when they know that most of the schools require both general GRE and then specific field GRE and then high score of TOEFL, things like that. I mean, right now, what I know from Rutgers is they didn't even bother to ask about your GRE, so you only need your TOEFL, and then it is a very good idea also try to communicate first with the professors, because most of the university have their own websites in which they put the department list and also included the faculty list. From there, you can understand which lab you are interested to take on, and then you can try to. They put their email address. You can try to send emails to ask if there is any available position for PhD. Yeah, that would be my tips that I can share with you guys.
Great tips. Anything to add from Marsha or Mira?
Okay, I will add. So first, I think you should do your research. So as he said, you have to be clear of like what you want to do. Is whether it's biology. No, sometimes it doesn't have to be like clear cut, but you have to know like the realm of like what kind of major that you want, and go for the list of university that you want to you get, right? And then make sure that you are actually try to fulfill every requirement, but it doesn't have to be perfect, because he's right. Although they mentioned that high rocket number of like TOEFL or GRE, but there are a lot of things I also consider. For example, like if you have like a very, very well-written personal statement that will actually knock their heart, it will actually compensate for other things that you might lack of. So it's not always like a math, it's not always like 1 + 1 is 2, but it can also like, maybe something weight more than the other one. So don't get scared or don't get hesitate or being hesitant when you think that your package is not perfect. Just have the courage to apply. I just want to encourage that you want to apply, because tip is that your authenticity matters, and that the fact that we are still underrepresented also matters, because they want diversity within the community. So if you're coming from a completely different background and you're coming and you're offering something, they will likely to approach you after you reach out, and that's true. When you want to reach out to the admission or you want to reach out to professor, they will likely reply, because this is such a general norm and a culture here where you actually communicate via email, even though you don't know them in person. And the last one, I would say, if you think funding is something that you'll be struggling upon, try to look for different types of scholarships. I think today there are so many different scholarships that are offered, both from the university itself and also from our home countries. So trust me, if you already do your homework, you already do research, you try to fulfill the requirements, there's a way that will open for you and doors will knock back to you. So don't worry, just have the courage to apply.
I think just a couple things to add. I think for me, the first thing is make sure this is what you want to do, like doing a PhD. It's a really long path. It's 5-6 years or even more. So I think the first thing I would say is make sure that's what you want to do. Because there are other paths, like you can do a master's, you can just work, you can do other things. For me personally, that there have been more than 1 or 2 times where I even considered leaving my PhD program in the middle, because it was just so hard and such a long way. So just make sure this is what you want to do. And if it is what you want to do, that's great. And I think, the way to figure out this is what you want to do is to get experience. So get research experiences as much as possible, like work in the lab or try doing research and see if this is what you want to do. And that will look really good on your application anyway. And if you do choose to apply, I think what Professor Arif said really echoed with me, that finding the right mentor is super important. Like finding a PI you can talk to, not just about science, but also even just your struggles. Like if you need something just for you personally, you can tell them, and that you feel comfortable to talk to them about anything really. Yeah, and I guess maybe lastly, just find support, like make sure you have like a good support system, and it's long, but it's very rewarding, I would say. Yeah.
Thank you everyone for the very helpful tips. So unfortunately, we are out of time. Again, thank you so much for all our speakers tonight. We really appreciate your time and willingness to share with us. I'm sure all of us can get something out of this discussion that might be useful for our future academic career or even life in general. I also want to thank all the participants for joining us, and PERMIAS Nasional AR&D team for organizing this event. We're doing another one in late February or early March, so don't forget to look out for those as well. And follow our social media at permias.nasional on Instagram and LinkedIn. And yeah, so I guess that's a wrap. Have a great day, everyone, and see you on our next event. Thank you.
Thank you.
Thank you, everyone. Have a good one.
Thank you, everyone.